Welcome back from break, everyone. We have reached our last session of the conference, and it has been such an amazing 2 days. Thank you all for being here with us. So for this last session, we will have Luke hosting a Q&A session with some of our therapeutic development collaborators based on their pre-recorded videos that we sent out before the conference. And while watching these pre-recorded videos, you probably saw that they are jam-packed full of new information about many different therapeutic approaches. And these presentations that we're discussing today, as well as all of the content we've discussed, are really important and very exciting. And one of our missions as an organization, as we're bringing you this content, is to make sure that every single person in this virtual space understands the truly remarkable progress that we are making in terms of therapeutic development. So if you've reached the end of the conference and are feeling a bit of information overload, because we certainly have thrown a lot at you in the past 2 days, that's okay. We have plenty of plans to follow up on all of this content and everyone's questions after the conference to help us all digest what we've learned this weekend. And so with that, I will hand it over to Luke.
Thanks, Dominique, and hello to everybody. Welcome back. Excuse me. This part of the conference is really exciting for all of us, and for me, it's an honor to be here and moderating this Q&A, because it's real tangible examples of all the progress we've made. And so I'm going to go through for the rest of the afternoon 4 of our collaborators who are working on hopeful therapeutics for our kids and for every individual affected by KIF1A-associated neurological disorder. So this is something that I'm incredibly honored to be moderating these questions. So hopefully everyone got a chance to look at the videos that were pre-recorded by each of these folks who spent a lot of time really laying out what the therapeutic strategy is and what they're working on. And this is a really important and great time to get some clarity and some answers to the questions that were sent in. And I'm going to stick to a lot of the questions that came in ahead of time, but I'll also try to keep an eye on the Q&A. So you certainly can be punching things in there. And if we don't get to things, we'll circle back with the folks and get answers to you. Like Dominique said, one of the things that we're so focused on is making all of this colloquial and relatable and understandable for everybody in our community, because everybody plays such a huge part of it. So with that, I'm not going to spend too much more time. I think I'll jump right into it and invite our wonderful friend, our collaborator, Omid Karkuti from Rarebase. Omid is the co-founder and the chief operating officer of Rarebase. And I can tell you that, Omid, all of your work and the team at Rarebase, what you guys are doing means so much to us. So thank you so much for being here. And thank you so much for recording that wonderful informative session. So hi, Omid.
Hi, Luke. Thanks so much for the opportunity. Really a great pleasure to participate in this with KIF1A.org and the CAN community.
No, it's so wonderful to have you here. I think maybe best we have 15 minutes, so I want to dive right into it. We got a lot of great questions following on that wonderful presentation that you were so kind enough to record. And the first question is one that we get all the time, and we get varying answers to it. So I think best coming right from you is, in your presentation, you talk specifically about an ASO that you're creating to treat or to address neurodegeneration associated with CAN and also other disorders. So first, I'm wondering if you could maybe explain to our community, what is an ASO?
Sure, of course, Luke. Yes, ASO stands for antisense oligonucleotide. It's in the form where we're applying it, it's actually a single-stranded special DNA molecule that's chemically modified to be resistant to degradation in cells. And it has been used in the past and has been the starting material and the type of chemistry around several different approved products, notably Spinraza for spinal muscular atrophy. That's an example of approved ASO product. And so in the way that ASOs work is the single-stranded DNA molecule can actually bind messenger RNA and inhibit its further translation because it causes those bound RNAs to be degraded by a pathway called ribonuclease H. So in a way, an ASO can be a way to design a molecule that can suppress the expression of a gene target, and we're doing that to try to pause or stop neurodegeneration for a specific gene target that we're going after.
That's really awesome. And I'm wondering, can you explain a little bit about how that ASO approach is applying to KIF1A and your strategy at Rarebase and the ASO you're developing?
Sure. So in CAND, we see both the spasticity that, and of course, KIF1A is also associated with what also referred to as HSP30 or hereditary spastic paraplegia type 30. So that spastic paraplegia phenotype, or that progressive spasticity, is one that would be a type of symptomology that could be addressed by this method, try to stop that progression. And then further, also, we heard from some of the others, there's neurodegeneration can also happen in the form of cerebellar atrophy. So there's the potential for, depending on the nature of the degeneration that is happening, there may be a way to slow or stop that disease progression, that part of the disease progression.
That's great, thanks. And you laid that out so clearly in the presentation too. A question that is on everybody's mind and that we always get, and I recognize that is really difficult to answer in so many ways, because so much of what we do is and what you're doing is based on real-time experiments. But assuming that this ASO continues to go well, as we certainly hope it does, and we see efficacy in the model systems, and by model systems I mean everybody, our cell lines and our animal models, etc. How and when do you think this could lead up to an actual clinical trial for CAND patients and for families to be involved?
Yeah, sure. I know everyone has the that's a goal for all of the programs that you all are sponsoring, and certainly for all of the disorders that we're really closely involved with and looking at. And so we're currently in the preclinical phase. We're in the phase of testing actually a series of synthesized designs in cells to determine their potential efficacy. And from that phase of screening candidates, we would move on to nominating a lead from those. And that is something that we would look to test in further model systems such as animals. If we have preclinical efficacy in cell-based models and animal models, then the stages that follow that would be safety pharmacology and toxicology studies that are required by the FDA prior to the beginning of a data to comprise a data package that's used to start a clinical trial. And then, of course, clinical grade manufacture would need to take place as well. I guess that if our studies are studies to continue to move forward with success, then the potential timeline to a clinical study could be between the 2 and 3 year time frame, pending success of those different types of animal studies and demonstration of safety in the preclinical models as well.
That's great. Thank you. That's exciting progress as always. And something that has been popping up a lot. We've been talking a lot about the importance of mitochondrial health in our CNS, in the nervous system. And I'm wondering if you could briefly address what mitochondria are, first of all, and how learning more about mitochondrial health can help us understand and hopefully treat KIF1A.
Certainly, yes. So mitochondria, they're commonly referred to as a powerhouse in cells. They're a subcellular organelle. There are many of them within cells, and they generate a molecule called adenosine triphosphate, or ATP, which is the primary unit of chemical energy or energy transfer in cells. So they're super important for providing energy, which is required for cells to live and do their job. And so in particular, if you think of cells in the central nervous system, they're more dependent on energy generation from mitochondria. They don't have, some other cells have a backup pathway for energy generation called glycolysis, and that's not really present in neurons, at least. And so there's a kind of an dependence on this pathway for energy generation. So it becomes particularly important. And example of that is there are many mitochondrial disorders, and you see the primary clinical presentation of the mitochondrial disorders. They're directly involving mitochondrial genes or genes that are transported to the mitochondria, and they present as central nervous system disorders. So it's very important to pay attention to energy as a resource for cells, as that's what's there to help cells stay healthy and do their job and stay alive. So there are a number of therapies really in development for mitochondrial disorders that are intended to correct abnormalities in mitochondrial function. But yeah, we're paying close attention to that, and that's kind of there also appear to be specifically, and as outlined in the presentation, abnormal changes to mitochondrial gene expression when KIF1A is suppressed, and that seems to have some effect on the viability of cells. So this is quite relevant specifically to CANT.
And something that isn't often discussed and that the community doesn't necessarily know about. So it was really, really incredible to see that and to see that laid out in your presentation. So thanks for addressing that. I also, something that is so vital to every therapeutic shot on goal that we're taking here are iPSC lines. And we hear that a lot. And I just think it might be really beneficial for the folks tuning in to understand what those cell lines are, what an iPSC is, and how it is the vitality of it as it applies to your process as Rarebase continues to develop these therapeutics.
Sure. Thanks, Luke. Yeah. iPSC stands for induced pluripotent stem cell. And these are cells that are genetically reprogrammed from fully formed somatic cells to be able to be more, let's say, embryonic-like. They become pluripotent in that, typically an adult cell doesn't continue to differentiate into, there's some cells that they're different, but they don't continue to change and turn into other types of cells. But of course, from an embryo, you're able to get all sorts of different kinds of cells, as you get every single different kind of cell in the body. You can artificially induce this state in a cell, and for example, starting from cells from the blood or cells from the skin, and turn those more into this pluripotent cell state. And with certain specific types of cell culture methods, these iPSCs can be cultured to differentiate into different types of cells, such as cells from the brain, cells from the heart, pancreas, liver, and so on. So there's different methods of cell culture that allow iPSCs to turn into different types of cells. In the context of KIF1A and CAN, we're really focusing on how we turn iPSCs into cells representative of, the brain or the spine. We're thinking of those types of cells, but they're super useful. And also, of course, KIF1A.org had previously made the investment of generating patient-derived iPSC lines, which we are currently using for our studies. And so that's it's not always that easy, as for many of the disorders we look at and work on, those resources aren't available. So that definitely saved us several months, like probably about 6 months to get work started. So that's a very nice resource that you all have invested in and made available.
Well, that's amazing. And it's a point I kind of want to hit hard just to everybody tuning in, is if you listen to what Omid just said, is that because of all of the work and the dedication and the participation of the families, we were able to collaborate with Rarebase to save 6 months off of the process. And so that is huge, right? All of the work that the families do, and by participating in research, I can't stress the importance of participation. We are able to have those cell lines for scientists like Omid and his team to go on and create those therapeutics. And you'll hear that throughout the rest of the afternoon. But a huge point to recognize is that it's because of the participation of all of you here in attendance of this conference that we're able to make this progress and save 6 months. A lot happens in 6 months to our kids. So it's just fantastic. Thanks for explaining that, Omid, and for pointing that out. It means a lot to us. Another question that came in is this is something that we briefly talked about before, but in the presentation you mentioned one therapeutic approach to CAN being upregulating gene expression, right? Can you kind of simplify what upregulation of gene expression means and how that could work as it applies to KIF1A mutations and the therapeutic strategy that we're taking?
Certainly. So in the context of a dominant or a heterozygous disorder like CAND, where there's one affected, one variant or one mutant gene copy, and we get a gene copy from each of our parents, from our mother, from our father. So we have pairs of homologous chromosomes. So there's 2 copies of the KIF1A gene. And so in although the variants, the mutations that arise in KIF1A are de novo, as in they're in the patient, they're not really typically inherited. It would be something where you look at upregulation. You have one good copy and one dysfunctional or mutant copy. Upregulation of gene expression would be to increase the amount of gene expression from one or both copies with the intent to compensate for the deficiency due to loss of half. So we might have loss of half of function, but if we could double the amount coming from the normal gene copy, that could compensate for the deficiency. And that really upregulation of gene expression specifically refers to increasing the amount of gene product or messenger RNA that can code for the protein and hopefully help to overcome the deficiency by the loss of one copy.
Thanks, Amit. I think that clarifies a lot, and it was something again that I hope everybody gets a chance to go back and watch more than once the presentation that you made. I do want to ask one more question because it's along the theme that we hear so much that rare is not really rare, right? We're looking at and learning from lots of different conditions. And in the presentation, you touch on that. You talk about having observed patterns between different neurological conditions, right? And I'm wondering if, what are some of those rare diseases that share common characteristics with CAND? And are you surprised when you find these characteristics and these shared commonalities? And is that going to speed up the process of therapeutics one day, hopefully?
Certainly. Well, since we started Rarebase about exactly 1 year ago, we've looked at, through the patient community, meeting families, meeting different nonprofit organizations, more than 120 different disorders that we've looked at. And about 2/3 of those are involving the central nervous system. And among those, some patterns arise, and they really have to do with, there's a series of these disorders, rare, ultra rare, and some common disorders that present with this spasticity or are actually classified as a hereditary spastic paraplegia. So that may be secondary, that may be the primary clinical presentation of the disorder. So we see some commonalities between phenotype, but we also touched on the kind of cellular energy and that bioenergetics. We see mitochondrial dysfunction. So I guess with respect to the type of therapies that we're developing, the neurodegenerative component that leads to that spastic paraplegia phenotype, we see common patterns or mechanisms of neurodegeneration that we are looking to address with a particular form of therapy that we're developing in collaboration. So that's one of the synergies. And then also further to that, the kind of as the root cause of a number of different genetic disorders, or the number of different genetic disorders, the root cause kind of converges on either that kind of neurodegenerative phenotype, or in some cases, the dysfunction around cellular energy production. So I think those are a couple of the common threads that we see, but of course, a series of disorders that has been really our company's focus are presenting with epilepsy, presenting with neurodegeneration, presenting with missed milestones or intellectual disability, and also with some autism or behavioral disorders. So that's been a pretty common theme and areas of unmet need in the field, is that the phenotypes, there's certain genes that lead to certain phenotypes, and then there may be ways to address those with medicines that are already there, or with medicines that we can discover and develop.
Yeah, thanks so much, Amit. And we're just so grateful to be both in your scientific wheelhouse, but also and I know how close our community is to your heart, just because you guys are constantly in touch with us. And I thank you for the presentation and for taking time to answer the questions that came in after the presentation. And I'll end with just asking you if there's a message to the community that you might want to share, or if there's something that we could do to help you on this endeavor. Please, now's the time. We have everybody together.
Yeah, I think for the CAND and KIF1A community, I would encourage you all to continue to attend events like this and participate in research. Like I said, the IPSC lines that we had access to is really because of that. And there's 7 of them. We're able to make use of 3 of the lines. One of them is particularly genetically very interesting. We don't really find those things. That doesn't happen unless people are participating and paying attention. You'll probably hear from the clinician scientists in the group around the natural history studies. These things are particularly important for the furtherance of clinical trials once the therapeutics become available. So I would say, for the community, I think you all have done a lot of really impressive things and made some tools and materials available that are kind of unique and uncommon, and that makes a difference. But continue to do what you're doing and participating in the research, and we love to see it. And if there are questions that come up we're easy enough to access, and Luke and Dominique and Kat, we're talking all the time, so we would be happy to kind of help think through some of those things if it comes up from the community too.
Omid, thank you so much, and just please do send our thanks to the entire Rarebase team. You guys mean so much to us, and thank you for being such a central part of our community and for taking the time. I really appreciate it.
Thank you, Luke.
Thanks, Omid.